This video will cover a couple of Hardy-Weinberg demo problems. The first thing we need to do when we start with a Hardy-Weinberg problem is identify the information that you are given for the problem. For all of the problems, you'll want to identify if you are being given allele information or allele frequency information, or information about the genotype or phenotype. Because if you are given information about a genotype or a phenotype, then it's very likely that you can figure out a genotype frequency and then work from there to get your allele frequencies. So just like any math problem, any math word problem, you'll want to look really carefully at the information you're given and the information that you need to find. For a really quick review of our variables, P is always going to be equal to the frequency of one allele. We'll say the dominant allele here. And that's going to be the number of dominant alleles divided by the number of total alleles in the population. If you have a problem with a different inheritance pattern, say codominance or incomplete dominance, you could also just think of it as the allele frequency for allele number 1. It doesn't matter too much as long as you keep track of it. But if you're doing a problem with dominant and recessive alleles, we are always going to make P the frequency of the dominant and Q the frequency of the recessive, just to keep things consistent. So Q is going to be the frequency of the recessive allele, and that's going to equal the number of recessive alleles divided by the total alleles in the population. Again, if you're given a different inheritance pattern, this could just be the frequency of allele number 2. For our genotype frequencies, P square is going to be the frequency of the homozygous dominant individuals. In other words, the frequency of, let's say, big A individuals. So this is the number of homozygous dominant individuals divided by the total individuals. 2PQ is the frequency of heterozygotes. And I'm running out of room, but that will just be the number of heterozygote individuals divided by the total. And then P squared is equal to the frequency of homozygous recessive. So that'll be the number of little a individuals divided by the total. A couple of things to keep in mind. If we are looking at the total number of alleles, remember that each individual has 2 alleles. So we may need to keep that in mind if we're counting out the total number of alleles in a population. If we have an individual who is homozygous, they're going to have 2 of the same alleles. Individuals that are heterozygous are going to have 2 different alleles, but they still have 2 total alleles. Also, you'll want to pay special attention to whether you're looking at alleles or individuals, because this will help you keep track of how many you need. So if we have a number of individuals, but we're counting alleles, we need to double our numbers. Whenever we are starting out on an allele frequency problem or a Hardy-Weinberg problem, we want to ask ourselves if we are given information on allele frequencies or genotype and phenotype frequencies. That will tell us if we're going to be able to start with P and Q, or if we're going to be able to start with P squared, 2PQ, or Q squared. Remember that P plus Q is always equal to 1. They're always going to be decimals less than 1. Same thing with P squared plus 2PQ plus Q squared. Each of those individual variables is a decimal that all add up to 1. In this example here, coloration in this species of scarlet tiger moth is a single gene, 2 allele system. So this is what we've been doing all along with incomplete dominance. Now, the type of dominance doesn't actually matter too much for our Hardy-Weinberg calculation because we have 2 alleles. One will be set equal to P, one will be set equal to Q. But it is helpful because the information that we're given gives us a total number of individuals. And it also gives us the total number of each genotype. So the total number in the population that are homozygous for the white spotted allele, heterozygous, and homozygous for the little spotting allele. This trait is showing incomplete dominance, meaning that individuals who are homozygous dominant have lots of white spotting. Individuals who are homozygous for the other allele, little spotting, are going to have just a little bit of spotting. And individuals who are heterozygous are going to have an intermediate amount of spotting. I know that these are not following the rules we've been using for incomplete codominant notations. Don't worry about that here. I actually don't mind because we usually say that P is equal to the dominant allele, Q is equal to the recessive, and that just kind of helps us keep track of that. So don't worry too much about the actual allele information here, or the allele notation, I should say. So we are, in fact, given genotype and phenotype frequency information. But we are also indirectly given allele frequency information because we are given a total number of alleles and a total number of individuals and a total number of individuals of each genotype, which means that we can calculate the allele frequency. So this particular problem is a little unusual in that we can calculate it from either direction. And I'm actually going to do that. I'm going to do it both ways for you. You are welcome to do it either way, whatever makes sense for you, but you should know how to do it both ways because depending on the type of information you're given, you may have to do one or the other. So the first thing that we need to know is the total number of alleles in the population. So we're going to do the allele frequency direction first, which means we're going to directly calculate P and Q. So if we have 1,612 individuals total, that means we have 2 alleles per individual. So in our population, we have 3,224 alleles total. For our individuals that are white spotted, we have 1,469 individuals. These individuals are all homozygous, which means that of those individuals, they each have 2 alleles. We have 2,938 big A alleles. I'm going to skip intermediate for just a minute. By the same logic, we have 5 individuals who are homozygous for little a. That means for these individuals, we have a total of 10 little a alleles. Now, the homozygous individual is a little bit different. They have 1 of each allele, which means in those individuals, there are 138 big A alleles and 138 little a alleles. So we don't need to do anything special with those because we know that each individual has 1 big A, each individual has 1 little a. So now we can calculate our allele frequencies directly. So let's say that P represents the big A allele frequency. and Q represents the little a allele frequency. Now it's just a matter of totaling our alleles. So P is the frequency of the big A allele, which is going to be 2,938 plus 138. So 2,938 plus 138 big A alleles divided by our total number of alleles, which is 3,224 alleles all together in the population. So this is 3,076 big A alleles divided by 3,224 total gives us the frequency of the big A allele as 0.95. We can calculate the Q allele one of two ways. The first way that I'm going to do that is by calculating it directly. So Q is the frequency of little a. I'm going to have to shift down just a little bit. This is going to be 10 A alleles, 10 little a alleles, plus 138 little a alleles, divided by 3,224 total, which is going to be 148 little a alleles divided by our total alleles gives us Q, which is the frequency of little a, 0.05. So now we have answered the question, total number, or I'm sorry, the allele frequency of P and Q. We could have also used the equation that P plus Q equals 1. And once we solved for P, which was equal to 0.95, we could then plug into this equation and get Q equals 0.095. For these problems, I'm generally going to say to round to 2 decimals. So no matter what stage you're in, keep it at 2 decimals. That way we're just kind of consistent throughout the class. If you round it too early, your numbers are going to get thrown off just a little bit. It's not necessarily that they're wrong, but you may come up with a different answer than somebody else if you do rounding, especially if you do rounding when you start squaring and square roots. So just go ahead and keep everything in your calculator. You can just keep the numbers as they are, but if you have to round, have to clear your calculator, use a decimal with 2 placeholders, and then at the very end give your answer in 2 decimal places. The other way to do this problem is to calculate the genotype frequencies and use those to get to the allele frequency. Now you have to be careful with this one because, as we'll see a little bit later, sometimes you don't have all 3 of your variables. So you have to pay attention to what you do have. For this one, we do have P squared. P squared is going to be the frequency of the homozygous white spotted allele. 2PQ will be the frequency of the heterozygous individuals, and Q squared is going to be the frequency of the homozygous for the little spotting allele. So for this one, I'm going to actually start with Q squared. Q squared is equal to the number of homozygous little a individuals divided by the total individuals, because this is a genotype, so we're basing it on an entire individual. So for this one, we have 5 little a individuals divided by 1,612 total individuals. So Q squared is going to be equal to 0.003. Now our question is actually asking for Q. So if Q squared equals 0.003, then the square root of Q squared, square root of 0.03, is going to give us our Q value, which is equal to 0.05. From here, we can find P by using P plus Q equals 1. We know from the previous page that when we do that, that's going to work out to P equals 0.95 using this Q value. We can also look at our number of homozygous dominant individuals. So P squared is the number of homozygous dominant individuals divided by the total individuals. So that's going to be 1,469 divided by 1,612. When we do that, we get, hold on, I'm just checking my notes, 0.9113. So P squared, 0.9, I'm going to say 113. I'm just keeping decimals for a minute, and then at the end I'll round to 2 decimals. So that means that P is equal to the square root of 0.9113, and that comes out to be 0.095. So either way, we have calculated our P and our Q. You have to be careful with this one though, because most of the time you don't know the difference between the homozygous dominant and the heterozygote. We know here because this is incomplete dominance and you can see those intermediate individuals. But in most of our problems, we cannot calculate based on the dominant phenotype. And I'll show you why in just a second. Our next problem is the simplest type of problem that you're going to get. Remember that we always want to look at what we're given in our problem. Are we given allele frequency information? Meaning, are we given the allele frequency, or can we calculate it directly? Or are we given genotype and phenotype information, or can we calculate it relatively easily? So this question, in a pea plant, yellow seed color is dominant to green seed color. If the allele frequency of the green allele is 0.3, so if you read this carefully, if the allele frequency is 0.3, we are given the allele frequency information, and we're actually given an allele frequency. We don't even have to calculate it. If the allele frequency of the green allele is 0.3, what is the frequency of yellow alleles? So let's go ahead and set our alleles. P is conventionally the frequency of the dominant allele. So we're going to say P is the frequency of the yellow allele. And Q is then the frequency of the green allele. So we're given the frequency of the green allele. So for this problem, Q is equal to 0.3. The easiest way to calculate P is going to be P plus Q equals 1. So P plus Q equals 1. So to solve for P, it's going to be P is equal to 1 minus Q. Go ahead and plug in that Q value, and we get P is equal to 0.7. So we've answered the first question. The frequency of the yellow allele is 0.7. We know from the problem that the frequency of the green allele is 0.3. Part B asks us, what is the frequency of heterozygotes? To answer this question, we need to look at our second equation. And in our second equation, we know that 2pq is equal to the frequency of the heterozygotes. So for our second part, part B, we are looking for the frequency of our heterozygous individuals, which is going to be equal to 2pq. So we can go ahead and just plug those values in. 2 times 0.7 times 0.3 is going to be equal to 0.42. So the frequency of the heterozygotes is 0.42, but what this means in a real population is that 42% of the population are heterozygous. For these questions, leaving it as just 0.42 is perfectly acceptable. That is actually answering the question. And I usually tell you to leave it, but sometimes the question is actually going to ask you what is the percentage in the population. And I want you to recognize that a frequency and a percentage are essentially analogous. But when we do the problems, you have to use the frequencies as a decimal, not as a percentage. When we're talking about the population, it's appropriate to say 42%, but in the problem, you have to use the decimals because we're always looking at a part of a whole. Our last problem is probably the most common type of problem that you're going to see. In this problem, we have 100 cats, and in this population, the tall allele is dominant to the short. So we're going to call the tall allele big T and the short allele little t. There are 84 cats that are tall, 16 that are short. So we're going to always go back to, are we given an allele frequency, or could we calculate it? Or are we given a genotype or phenotype frequency? In this case, we are given phenotypes, which means we can infer the genotype. So for an individual who is short, their genotype is going to be little t. And we know that there are 16 short cats. For an individual who is tall, they have two options. So tall individuals could be big T or big T little t. We don't know, based on this information, which ones are homozygous and which ones are heterozygous, but we do know that there are 84 tall cats. So before we move on, let's just look at our genotype frequency equation. P squared would be the frequency of big T cats. 2pq is going to be the frequency of heterozygous cats. Now, we cannot tell the difference. All we know is that there are 84 total cats who appear to be tall, and they can be either one of these genotypes. So what we're really saying in here is p squared plus 2pq is equal to 84. We are not going to solve a problem for two variables in this class. It's not even, for the scope of our class and the math that you're required to have, it's not even possible to do that. So we cannot set 84 equal to either of these values because it's equal to the sum of p squared and 2pq. But we do know that q squared is going to be the frequency of little t. And we do know how many individuals are that genotype because the short genotype is represented in these 16 cats. So all of those 16 cats are homozygous recessive. So for this problem, we're going to start with q squared. Q squared is going to be the frequency of the little t allele. So this is going to be 16 short cats divided by 100 total. So q squared equals 0.16. And we can take the square root of both sides to get Q, which is going to be 0.4. Now, we cannot solve for P directly from our 84 total cats. You could do some crazy math and solve for it now, but let's just use our easy equation, P plus Q equals 1, which means if Q is 0.4, then P is 0.6. So the frequency of the Q allele is 0.4, frequency of the P allele is 0.6.